Great. Thank you all. Thank you to all the organizers for giving me a chance to speak today and tell you a bit about what we're doing in the Lay Lab. As Lisa mentioned, I'm focused on methanogens in the gut microbiome, but actually today I'm going to talk to you about a different project that involves a broad look at host microbe associations. And this is assessing basically all of vertebrate taxonomic diversity. And during the course of telling you about this project, I'm going to try and highlight some aspects of bioinformatics research that we're doing in our group. So how do humans and all animals acquire their unique microbiomes that we observe, say, in their guts? And given that animals evolve in a microbial world, one could imagine that during the course of evolution, symbionts are gradually acquired, and this can result in a pattern of phylosymbiosis, where microbial communities of closely related species look more similar. So this lizard and chicken in this example, microbial communities would look more similar than to the monkey, and all three would look more similar than to the frog or fish. And this can often be measured by a beta diversity, for instance, Bray-Curtis, comparing all microbial communities, and can be represented here as a dendrogram, showing microbial community similarity. And one can see if there's congruence between the host phylogeny and then microbial community similarity. And if there is a congruence, then that suggests that host evolutionary history plays an important role in microbial community assembly. This contrasts to an alternate scenario of environmental uptake in which ecological processes really dictate microbial community assembly, such as host diet, host habitat, or host geographic distance. And here, microbes are uptaken from the environment or other hosts relatively rapidly. And given that this is done on ecological instead of evolutionary scales, one is likely not going to see this congruence between microbial community similarity and host evolutionary similarity. So one can use these data to try and identify what are the main factors for microbial community assembly. And so that was the goal of our research here. And this is in a collaboration with researchers from Vienna, and they were able to collect a very large and impressive data set of animals collected from around the world, shown in this map with these red dots. And then the samples, the gut samples were, or feces samples were processed in a very standard manner with 16S sequencing. Here's a breakdown of the data set on the right here in terms of number of samples and number of species for the 5 host classes, taxonomic classes, with ray fin fish, amphibians, birds, mammals, and reptiles. And you can see that it is biased towards mammals, but we did try and at least include non-mammals since they really have not been studied well. And importantly, the coloring here shows which samples came from captive versus human versus wild samples. We did include a few humans as references, but as you can see, most samples are from wild individuals. And this is important given that captivity can greatly change the microbiome relative to wild counterparts. So wild animals really haven't been studied to this extent, and really no study has investigated this much taxonomic diversity too, especially with wild animals. So here is the whole data set in terms of the animals included. This is a super tree with the classes mapped on with the various colors. So again, fish, amphibians, reptiles, birds, and mammals. Hopefully the silhouettes of the animals on the outside gives you an idea of which animals are where on the tree. I recommend taking a second to look over the tree. I will show it quite a few times during the course of the study. Given that the mammalian clade is so big, I'll just point out some important clades, such as primates here down below, and then a little farther over, you have rodentia, and then hindgut fermenters, such as horses, and then carnivora, and then up near the top, you have ruminants. So one great thing about this data set was all the metadata collected with it, and that's useful for looking at host evolutionary history versus other components that could dictate microbial community assembly. So for instance, here's the same tree as before, but now here is host diet mapped on, at least in very broad categories of herbivore in green, omnivore in orange, and carnivore in red. But beyond that, we had detailed diet information shown in this heat map. So the dendrogram is showing basically listing the different detailed diet information, such as leaves, grasses, roots, but also vertebrates and invertebrates. Yellow is showing that the animal did consume that food versus black did not consume. You can see some are in orange, and that represents 0.5. That's an average across individuals of that same species. For instance, we had a few giraffe individuals, some were captive, some were wild, and so they had different diets. And so this is showing the average. The dendrogram is just ordering them by their distribution across the animals. So beyond diet, we also had habitat information in terms of anthropogenic, aquatic, terrestrial, and cultivated terrestrial environments, but also captive wild, as I showed before, and sample type. Some were feces and some were actually gut content samples. And that's shown in these bars around the tree. For instance, some bars you can see are a mix, and that's because multiple individuals per species. And so some were, for instance, captive, some were wild. So a pretty impressive data set. It did have complexities such as a bias towards mammals, different numbers of individuals per species, and different metadata per individual. So it was a bit challenging to analyze. But the main question was looking at phylosymbiosis. And so a common approach used here is to just create some ordinations of beta diversity, here unweighted unifrac and weighted unifrac. And then the samples just represent the individual samples for the whole data set with distance representing beta diversity dissimilarity. So how similar the samples are. The samples are colored by host class in this case, so you can get an idea of whether there is clustering by phylogeny or at least taxonomy here. So you can see some clustering by mammals and maybe a bit of clustering by birds. But if you color the samples by diet, so you can see some clustering, by herbivores here in green, or say carnivores in red. So what is the most important factor here? Evolutionary history or diet? It's kind of hard to tell. So a more detailed analysis is really necessary and more quantitative. And to do this, I used multiple regression on matrices. As it sounds, it's multiple regression, but here on distance matrices. And use this approach because phylogeny can be directly represented as a distance matrix of branch lengths or patristic distances. Other factors such as diet, especially the detailed diet information, can be converted to a distance, say with Gower's distance, which is a pretty generalized distance measure. So then say host phylogeny can be compared to beta diversity to see if there's a correlation, and this can be done with diet and other factors. So here's the results, at least in regards to looking at unweighted unifrac for beta diversity, and the factors were host diet, geography, habitat, phylogeny, and technical. Technical included sample type, whether it's gut contents or feces, and also whether the animal was captive or wild. And so you have a coefficient or the effect size, and then the p-values. The box plots represent a way to deal with the different number of samples per species. So this varied depending on the species. And to deal with this, we just did a subsampling approach where we created 100 subsamples of one sample per species, and then looked to see how various sampling affected the results. So a pretty basic approach, but gave an idea of how robust the results were to the intra-species variation, within species variation. So in terms of the associations, the strongest was with diet, and then phylogeny, and then the others. And the only two that were significant were diet and phylogeny. Diet was stronger than phylogeny with unweighted unifrac at least. What about weighted unifrac when abundances, microbial abundances, are taken into account? On this case, diet and phylogeny were still the strongest association, but only diet was actually significant, where phylogeny was not quite significant. So these results, plus others I won't show today, suggested that diet is actually a stronger factor for microbial community assembly in these vertebrates, but phylogeny is important too. So there was a significant signal of phylosymbiosis, as I showed, even while accounting for other factors such as geography and habitat. A challenge with phylosymbiosis, or downside, is that it's kind of a coarse measure because you're looking at the whole microbial community together, and just using beta diversity, and it can really depend on the beta diversity measure assessed. So another approach that one can use is cophylogeny, to look just to see, if you've got, say, with a particular microbe, do you have a corresponding phylogeny for that microbe versus the host? And so you could get the phylogeny for the microbe based on 16S, RPOB, or another phylogenetic marker, and then just use a test to see if they're corresponding. So this is a bit more resolved than looking at the whole community. It gets a little bit more complicated because you can actually do cophylogeny for the entire microbial community at the same time using the Procrustean approach for cophylogeny or others. So the advantage here is that you can look at the whole community, but then you can also parse the results, as I'll go into here in a second. Just to briefly explain this, you've got the host phylogeny for this analysis and the microbial phylogeny. Here I'm using 16S phylogeny. These can be represented as distance matrices of branch lengths. And the third component is an incidence matrix of which microbes are associated with which hosts. And so this provides a global test of fit between the two phylogenies, but also you can look at global fits of particular host clades with particular microbial clades. So this is a nice advantage over, say, standard phylosymbiosis approaches. So I use this approach for our data set, this PACO method, and the global fit was significant. And as I said, you can look at local fits, and the strength of fit is based on Procrustean residuals. Larger residuals mean a worse fit. So worse fit, or just the residuals were parsed by host clade. So ray finfish, amphibians, reptiles, birds, and mammals, and then the diet here in the colors. We saw an interesting pattern in terms of the fit, where the fit was actually worse for ray finfish, and then a little bit better for amphibians, about the same for reptiles and birds, and then much better for mammals, especially herbivores. So other data did support this. It didn't seem to be a technical artifact of this analysis. And this is a really interesting pattern, since you see that mammals really have the strongest association, the strongest cophylogeny signal, especially herbivores. And this could be due to the fact that mammals, and especially herbivores, have very complex GI tract morphologies, but it also could reflect increasing complexity of adaptive immune systems, or maybe the social structures and placental birth in mammals. So more analyses are needed to really assess this, but very interesting pattern that we observed. So we could go even further than this, especially given the large data set that we had, and look for specific microbe-host clade associations. To do this, I used a method of phylogenetic signal, which basically means just clustering of a trait on a phylogeny. And so that's kind of represented in this example here, where you have a particular host trait, sensitivity to pollution in this case. And then you can see it's clustered, where you see, similar similar animals in this case show similar traits, and that's stronger clustering than just random distribution or simulation of Brownian motion traits, trait evolution. And so I use this approach for our data set, and here are the results. In particular, the analysis was called Local Indicator of Phylogenetic Association, or LIPA. And so here's the host tree, as shown before, but it's just turned on its side. You got mammals down here, and then non-mammals up above. Host diet is mapped on for reference. And then up here are the microbes that showed a significant phylogenetic signal. these are particular operational taxonomic units, or OTUs. It's mainly firmicutes and Bacteroides. And as you can, so the heat map, just in general, shows where you had significant host clade associations. The darker, the stronger, and white means no significant association. So you can see here that there's no associations with non-mammals, or with, yeah. So it's only associations with mammals. And this reflects the PACO results, where mammals showed a much stronger signal of cophylogeny. And so there was a distribution of associations, for instance, firmicutes here, various clades with primates, or with various carnivores, or here with ruminants, especially ruminants and especially cattle. And this is summed up here on the left. This is grouping by host taxonomic order and each microbial phylum. You can see it's mainly firmicutes across, with some Bacteroides, and it's especially prevalent within ruminants. So ruminants were quite special here, and they do have arguably the most complex gastrointestinal physiology, which might be causing this strong signal. So that was looking at host microbe cophylogeny and phylosymbiosis, but as I showed previously, diet was also a strong factor, so I want to look at that in more detail. But there is an issue of looking at diet in the fact that phylogeny kind of gets in the way, because phylogeny, phylogenetically similar microbes, or sorry, hosts, phylogenetically similar hosts generally have similar traits just because they have a close evolutionary relationship. So it helps to account for host phylogeny when looking at trait-trait correlations. As shown here on the right, this method is phylogenetic generalized least squares. It's often used to account for phylogeny when looking at trait-trait correlations. And so here in this example, it's looking at whether body mass trait and cerebellum mass is correlated, while accounting for host evolutionary history. And so I did that with host diet as trait 1, and then various aspects of microbial diversity. And so again, I was looking at whether there's association between diets and microbes while controlling for host phylogeny. And when I looked at an association between host diet and alpha diversity, I did see a strong association relatively. So here's the effect size and then p-values. The box plots represent that subsampling approach as I showed before, but both of these alpha diversity measures were significant, Shannon and Face PD, and yeah, the effect size was relatively strong. And when I looked at diet microbial taxa associations, individual microbes, I didn't actually see many associations, and there's actually just two OTUs here. So these results combined, plus other data that I don't have time to get into today, really suggested that diet more selected for total microbial diversity and not particular microbes. And this contrasts with host evolutionary history, where it's more really selective for particular microbial clades. So diet and host evolutionary history dictate microbial community assembly in the gut differently. And that was an interesting aspect that came out of these data. So in summary, we did detect a phylosymbiosis pattern, and that was mainly using multiple regression on matrices. And that did account for other factors. So it really does appear to be a true association, at least based on the metadata that we were able to collect. We did also see a significant cophylogeny pattern, as with the PACO approach, and it was interestingly strongest for mammals and weakest for reef and fish. And this pattern, as I said, might be due to a number of different factors, such as gastrointestinal tract morphology, complexity, and really it'll be great to follow up on this and see what are the main factors causing this pattern of cophylogeny. And then Diet was actually a stronger factor than host phylogeny, but it did seem to select for total microbial diversity versus particular microbes. So different aspect of microbial diversity was more dictated by diet. And this could be due to just, a selection for the particular functions necessary to degrade or to break down that diet. And necessarily, it might not depend so much on which particular microbes are performing those functions. So that was with all 16S data. And the nice thing about 16S is that, it's relatively cost effective, but it does only provide information about taxonomic diversity. Whereas shotgun metagenomics is becoming cheaper and easier, and it provides a lot more data that one can use. The problem is it also is a lot more complex to analyze. But we went ahead with this for the animal gut data set. As shown here, we actually expanded the data set in terms of the number of samples and number of species included. We tried to make it a little bit less mammal bias here, and we kind of succeeded in that regard. And we were able to try and balance a bit more in terms of number of samples per host species. But we did run into some problems, or some challenges, I should say. And that came into play right with metagenomic profiling, which is the basic method of assessing microbial diversity with metagenomics. And the approach is basically take Illumina reads, or if you have long reads, quality control them, and then classify the reads with an approach such as Kraken-2, using often a default database that comes with the tool to get the taxon abundances. So Kobo, a PhD student in our lab, looked into the issue of the fact that for even human gut metagenomes, often the percent of reads classified was relatively low using these default databases. So here, for instance, with Kraken, you get less than 50% on average classified using the Kraken reference database with human gut metagenome samples. It didn't get much better using this other database of collection of microbial genomes. So Kobo and I created Struo. It's a tool for creating custom databases, and it leveraged the genome taxonomy database, which I highly recommend everyone uses for microbiome work. It has a standardized microbial taxonomy, unlike the NCBI taxonomy. And this standardized taxonomy helps out quite a bit for creating a robust taxonomic assessment of microbial diversity. So we integrated that into Struo in this somewhat complex pipeline. And the pipeline generates Kraken and Bracken-2 databases for taxonomic profiling, and also human-2 databases for gene and pathway metagenome profiling. And so using that, we're able to show that the GTDB custom databases substantially improve the percent reads classified for both Kraken and Bracken, also a bit for human-2 at the gene and pathway level. The problem here is that we quickly face that the GTDB is rapidly expanding. So we created Struo at the release 89 version, and it's now on 202. And you can see how much it's increased in terms of the number of genomes, gone from 1,000 or 150,000 to 250,000 for number of genomes, and number of species, it's basically doubled. So our pipeline did not really scale that well to handle this massive increase in genomes. So while this is exciting that we're gaining so much data in terms of number of genomes that are publicly available, it's also more challenging to deal with. So I tried to address this by creating a more efficient version of Struo. And just to briefly explain that, basically I shifted the algorithm to rely heavily on MMSeq2. I won't go into details here, but using this approach, I was able to speed up the pipeline quite a bit, as shown here on the right. This is just checking the scaling of both approaches, Struo 1 in red and then Struo 2 in blue. And for the number of genomes, you can see the CPU hours needed is a lot less for Struo 2 versus Struo. And this was not at the sacrifice of sensitivity. You're showing the number of annotated genes. This is the same for each pipeline. And a nice advantage of the new pipeline was that with MM62, it could iteratively update the database. And that could save between 15 and 20% time relative to not using this iterative approach. So that can greatly help as new versions of GTDB come out. So that was a bit of an aside, just to highlight some bioinformatics work that we're doing. But getting back to the animal gut summary or data set here, this is the summary as I showed before, and this is the phylogeny of all species, much like the phylogeny that I showed before, but does include more species now. We tried to get a more even coverage across animal clades. And what's mapped on out here is the taxonomic profiling that we did. But this is at a broad level in regards to first thing that I dealt with was reads that map to host, the host genome. So depending on the sample, actually, there was a lot of host DNA included. And I tried to identify that using a number of different custom databases. Most reads map to the vertebrate host genomes that I was able to utilize for this. But I created a number of them, including just all vertebrates, plants, fungi, protozoa. And then, as you can see here in red, that for some samples, a lot of the reads were actually classified as host. And this corresponded with the fact that many of these samples were gut content samples instead of feces, and they contain more host tissue and more host DNA. In terms of other non-microbial DNA, you could see here in dark green that some samples, especially from herbivores, did have a relatively high amount of plant DNA, but it wasn't too common. Now in purple, that's the bacteria and archaea classified based on our custom GTDB database produced with Struo. So you can see it varies quite a bit depending on the particular animal species, and it can vary even between relatively closely related animals. And so the gray here on the outside is unclassified. You can see that for some animals, we were able to classify quite a lot, and others not very much at all. So this is a big challenge for us. A lot of host reads for some samples, and then a high variability in terms of the percent classified as bacteria and archaea. This lack of reads classified indicated a lot of novel microbial diversity that could be obtained using metagenomics, and that's what we went for next, using metagenome assembly to try and assemble genomes that were novel, that haven't been seen before, because these animals have not really been assessed in terms of their gut microbial diversity. And so I created a metagenome assembly pipeline developed over a couple of years. And here's a simplified version that I won't really go into, but the outcome is to produce genomes from the metagenomes. And these are called metagenome assembled genomes, or MAGs. And so with that, we were able to produce an okay number of MAGs, about 300, and almost all were unique species. Here's a breakdown of the number of MAGs for each class. And then it's parsed by whether the MAGs were taxonomically classified to an existing known species or not. And you can see that actually most of these MAGs were not classified at the species level, suggesting taxonomic novelty. These are new species. So based on those results, and they were encouraging in terms of the taxonomic novelty, I expanded the data set to 14 other vertebrate gut metagenome data sets that had not been used for metagenome assembly, hoping that I could expand on the novel microbial diversity that could be assembled. And so it worked quite well in terms of number of MAGs generated. That's shown in this bar plot. The number of MAGs is the number above the bars. The number of samples included was below the bars. And I really tried to focus on animals that have not gotten a lot of attention in terms of microbial gut diversity. So tried to stay away from, say, cattle or mice. And so in total, I was able to generate about 5,600 megs that were non-redundant, and that comprised of over 1,500 species. So here's a breakdown of those megs in terms of numbers of them per class as before. You can see quite a few more, the most being Clostridia and Bacteroida. This taxonomy, I should say, I've switched over to GTDB here, which is why it might look a little different for you. For instance, you have Firmicutes A, Firmicutes B, etc. Sorry for not pointing that out already. From now on, it will be GTDB. Some taxonomy matches the standard NCBI, some does not. But just want to point out here for the parsing of species level hit or no hit, most again were novel at the species level, suggesting a lot of novel taxonomic diversity. And I should say, just as one more aside here quick, you might say, well, how good are these megs? You know, the problem is you're assembling them from metagenomes, and people know that they're incomplete and you could have misassemblies. And as a quick aside, I want to say that we have been working on this issue. For instance, we've developed a tool called Deep Maze, which uses deep learning to try and identify metagenome misassemblies. This was a collaboration with members from the MPI for Intelligent Systems right next door, with Matteo and Bernard, and from ETH Zurich with Olga and Gunnar. And just real quick, the approach just takes the contigs assembled from a metagenome assembly, and then you map reads back onto the contigs. And this is a standard approach used for many analyses with metagenome assembly. So, and just with these reads mapped onto the contigs, the deep learning approach tries to identify misassemblies. The ground truth here was mainly simulations. We used simulations because they could generate very realistic data for this task. But we did do some validation on some real data, and the results were very encouraging. So this is a comparison of Deep Maze to the state of the art. This is a precision recall curve, and you want, farther top right is best. Random is shown here in this dashed line. The state of the art using two different metagenome assemblers, MegaHit and Metaspades, shown here, and then our approach with those two assemblers is shown over here. And you can see the Deep Maze substantially improved upon the state of the art. So yeah, that was just an aside to talk about a bit more of the bioinformatics we're doing. We do have other deep learning projects going on, and we're looking for collaborations for more in regards to deep learning and machine learning in general. So let me know if you're interested in potential collaboration on microbiome and metagenome data. But back to the animal gut metagenomes. So as I said, we had this problem of metagenome profiling, where a lot of samples did not get a lot of reads classified as bacteria and archaea. But then we did metagenome assembly to produce a whole lot of megs, many of them taxonomically novel. So we created a new custom database with all those megs included, and we looked to see how much that improved the percent of reads classified. And it did improve. Some species were big winners, others generally not so much of an improvement. The biggest improvement in terms of percent increase of reads classified was for mammals. And that makes sense given that a lot of the megs we generated were from mammals. So it did help to do all this metagenome assembly in terms of improving taxonomic profiling. But now that we had all these megs, we definitely want to analyze them further. So here is a phylogeny of all species level megs, all 1,500 approximately, with the phylum level taxonomy on the outside. It's mainly Firmicutes A, which holds Clostridia, for instance, and Bacteroidota, which is the name for GTDB in GTDB. One thing we want to assess here was whether these megs were really host specific or maybe more environmental specific, given that just because you are able to assemble a meg from a host gut metagenome doesn't mean that it's really host specific. it might just be transient in the gut and actually more prevalent out in the environment. So to do this, we collected a publicly available collection of metagenomes, both from environmental sources and host associated. And then we identified the abundance, or determined the abundance of each of these species in terms of these metagenomes to determine which are host associated versus which are environmental. And we look for enrichment using the standard approach of DESeq2. So this is just assessing which ones are host associated and environmental. Blue is showing which ones are significant for each particular phylum. Red is not significant. And you can see that most were significant for hosts, regardless of the phylum. And this was to be expected since we did assemble these from host gut metagenomes. But we did identify some that were actually significantly enriched in the environment. And these were from proteobacteria and actinobacteria. And then there were a few in these same clades that were not enriched either way, with a couple from Firmicutes A. And so we went further in terms of this host environmental split. So after identifying these particular species, we want to look at what traits were enriched in these host enriched species versus environmental enriched. And so based on the genome content of these megs, we were able to predict these 67 different traits using machine learning. And then this is just a breakdown of those traits, how prevalent they were in the environmental megs, in the host megs, and in the megs that were shown here in red, that were neither significant in either biome. And so you can see here for the host megs, as you might expect, they were significantly enriched in the anaerobe traits, so mainly anaerobes. And actually, you saw the opposite for the environmental megs, where down here, they're significantly enriched in aerobes. The environmental clades were also highly enriched with catalase trait and biosusceptible, suggesting they're not really adapted to the host gut environment. Whereas for host megs, they could grow at a higher temperature, but also various sugar consumers. So we're trying to get at what adaptations are, what phenotypes are selected for these particular biomes, the environment biome and the host associated biome. We also looked at just general diversity in terms of genomic diversity. A particular trait that we wanted to assess was biosynthetic gene clusters. We're interested in this because, as I mentioned before, not a whole lot of work has been done in terms of microbial genomic diversity in many of these animals. And so we identified biosynthetic gene clusters with anti-SMASH, and this shows the breakdown of them. In total, we got about 2,000 BGCs, and most appeared to be novel. And so you can see the most abundant ones across megs were non-ribosomal peptide synthetases, but then you do have a distribution of quite a few of others present among these megs. And just another way to plot that out here, you have the BGC type on the y-axis, and then on the phylum level clade on the x-axis, and then number of BGCs as circle size. And this just helps show that some BGCs are quite abundant and prevalent across various clades, while others are more specific towards individual microbial phyla. You have a wide diversity of associations between BGCs and phyla. So we looked at this in more detail in terms of the species that had the most BGCs. Here, just showing species with at least 3 BGCs, which is 233 shown in this phylogeny. Orange dots represent good bootstrap support. There's a lot of data on this tree, so I'll go through it one at a time here. The first ring is taxonomic association in terms of phylum level taxonomy. And the main clades here are actinobacteria, Firmicutes A, proteobacteria, and Bacteroidota. But we did have a whole lot of taxonomic diversity shown here in the legend. And then the second ring here is the taxonomic novelty, where the blue means there's a species level match, red means no species level match, novel species, and then green is novel at the genus level, so even more taxonomically novel. You can see it varies quite a bit along that ring. And then the third ring here is showing host environment enrichment, as I showed before, with blue being environmentally enriched and red being host enriched. The next ring is the prevalence of the major BGCs across our animal gut microbiome data set, the mammals, non-mammals, all those metagenomes, which BGCs were most prevalent. And you can see here that the RIPs shown in blue were actually most prevalent, especially these RIPs found in proteobacteria, this clade. And then the final ring here is the number of BGCs per species here. So you can see there's a number of BGCs shown in various colors. You can see some phylogenetic clustering of BGC types across the tree, and many don't have a whole lot of BGCs, but you do see particular clades with a lot, as Streptomyces down here in Actinobacteria, as you might expect, and then Proteobacteria. Interestingly, both of these clades were actually more environmentally enriched, not host-associated. The host-associated species with the most BGCs was down here, actually, in Firmicutes A. And you can see that it has the biggest spike. And I believe this was a Clostridia species. So yeah, the MEGs were very useful in a number of different ways, but a limitation of them is that you do need a large amount of sequencing data to get enough coverage to assemble them. So we did not cover the total diversity by any means. So we used a different approach to try and get at more of this diversity, and that was a gene level assembly. And that was done using relatively new tools by the same creators of MMSeqs, particularly PLOS for gene-based assembly, and then LinPlus for clustering those genes. So I applied this approach to the entire animal gut metagenome dataset and generated a dataset of over 150 million non-redundant gene sequences. Just as a reference, other studies of, say, pig gene gut collections or chicken gene gut collections, they range on the area of 5 to 10 million. So quite a bit more gene sequences generated here. This is a taxonomic distribution shown here in this plot. And this is just showing the genes after clustering at 50% identity, which shrunk it down to 6 million gene clusters, but still quite a bit considering that's 50% sequence identity clustering. You can see most of these clusters, almost 2 million from Proteobacteria, large amount from Firmicutes and Bacteroides. But we did also obtain some genes from viruses and archaea. And this is only showing the top taxa, but actually over 80 different phyla were represented. This could mean that, taxonomic classification was not perfect, and some of those could have been errors, or it could hint at a lot of unknown diversity in the gut microbiome. Based on some other data with archaeo targeting 16S primers, it does seem like there's a lot of diversity that we have not really assessed, because a lot of assessments of animal gut microbiome diversity has been biased towards bacteria and not really looking at archaeoclades, probably biased also to certain bacterial clades. So metagenomics provides more of an unbiased approach to really assess the diversity. And so we looked at this gene diversity in a number of different ways and characterized it, such as with Cassie family diversity, and we found quite a lot of diversity, suggesting there is, a lot of gene diversity here that could potentially be mined for biotech purposes, along with the BGCs. So for the top hits, for instance, we had a glycosyltransferase and a couple of glycoside hydrolases, and these particular families suggest a high amount of metabolism for peptidoglycan, amylose and other sugars, and chitin. But then you can see we have a huge amount of other diversity present. So we also did an analysis much like we did with the MEGs of host versus environmental enrichment, but here at the gene category level. So we grouped them here by COG categories and then looked which COG categories were enriched in the environment or enriched in the host. And this is parsed by COG category per phylum. And interestingly, so the circles represent basically the strength of enrichment, and only significant enrichments are shown. So, sorry, one second. So you can see here that we had a strong enrichment for all COG categories almost in the environment for proteobacteria, and mainly for methanogenic archaea, euryarchaea. But say here in the middle for verruco microbia, we saw some COG categories enriched for the environment, while other COG categories enriched for host-associated, suggesting that within this phylum, certain taxa with certain COG categories are enriched more in the environment, while others are host-associated, kind of a split within the phylum level. But interestingly, for certain phyla, it really was enriched in just the environment or in the host. And it was all COG categories, like Bacteroides and Firmicutes here. And this suggests that, well, the same functions are going on in both environments. Both environments are selecting for these functions. It's different taxa that are mainly doing it, especially for, say, proteobacteria versus Bacteroides and Firmicutes. And this was seen using a number of approaches, such as also assessing KEGG pathways. It's turned on its side just for room, but basically the same thing where you have environment and host-associated phyla down here, and then KEGG pathways. And just as before, you have environmental enriched KEGG pathways for these major pathways for, say, proteobacteria and euryarchaea. But for host-associated, then, we saw those same enrichment of, say, Firmicutes, Bacteroides, Tenericutes, etc. So this was an interesting finding, suggesting that there is a decent amount of functional redundancy, at least at this broad functional level of KEGG pathways or COG categories. So similar functions, but different taxa doing them, depending on the biome. So in summary, we saw a lot of novel taxonomic diversity, and this was kind of expected given how little most animals in this data set have been studied, especially wild animals. That was also a problem due to the trying to do metagenome profiling, since for many of the samples, only a few percent of the reads were actually classified as bacteria and archaea. To deal with that, we generated a lot of MEGs to try and bolster up the database. And we also analyzed the MEGs and found that most were host-associated, but some were actually not. So some did appear to be transient, and the traits between host-associated and environmental-associated MEGs did differ. And that gave us kind of an indication of the adaptations required for living in the host biome versus the environmental biome. And we did see a lot of genomic diversity among these MEGs, such as biosynthetic gene cluster diversity, especially RIPs and non-ribosomal peptide synthetases. And this, could have implications for bioprospecting and, bioprospecting new biosynthetic gene clusters for natural product discovery. And more generally, we saw quite a lot of gene diversity when doing gene level assemblies. And interestingly, based on our host environment association analysis, we did see potentially a lot of functional redundancy where, it's the same COG categories, same KEGG pathways, but different taxa performing these processes in these different biomes. So for future directions in this regard, we'd still like to go forward with the metagenomics further, especially now that we've created a larger data set for assessing diversity. And so I'm interested in looking at species level phylosymbiosis and cophylogeny. You can do this much more robustly using MEGs and metagenome taxonomic profiling. And I'd like to look at phylosymbiosis at the functional level, since you can look at genes and pathways with metagenomics. And also like to look at more diet function associations. As I said before, diet seems to be selecting for function and total microbial diversity versus particular taxa. And more generally, like to look at adaptation to the gut environment, by assessing or inferring host or microbial phenotypes based on genome content. So that's basically it for that project I want to talk about today. But I just want to give a quick plug for the Bioinformatics Working Group. I run that, and we meet every Thursday at 4 p.m. on Zoom, every other Thursday, I should say. We would have met today, but this workshop was more important. So it'll be 2 weeks from now is the next one. Anybody's invited. It's not just MPI, and it's not just microbiome researchers. And we cycle between a journal club on bioinformatics paper, a lecture series on bioinformatics topics, and then just a roundtable discussion talking about bioinformatics issues that we're all having. So with that, I would like to thank the Lay Lab and our collaborators. And within the Lay Lab, I'd specifically like to thank Silke, our technician who generated the next generation sequence data, Tony, a postdoc who helped out quite a bit initially with the animal gut 16S data set, and Jacobo with the Stro data set, and then Ruth for just general guidance and support. And I'd like to thank you all for listening today. I'd be happy to take any questions.